Okay, and we are now live. Yeah, first I'm gonna get some admin things out of the way. Hi, everyone. You can ask questions in the chat and I will collect them and ask them to the speaker later. For this, you do need a YouTube account. We are thinking of different solutions to approach this, but for now, make a YouTube account and ask the questions this way. And then also after the talk, there will be a chance to talk to Eugenia in the Zoom. So for that, after the talk and the questions are over, I'm going to post a link. Hold on. Okay, good. Yeah, and then obviously the whole talk will stay online on YouTube and you can watch it whenever you have time. Now I'm really excited to move on to introducing our speaker, Eugenia Chiappe. She did her undergrad in Argentina, and then she moved to the US to do a PhD at the Rockefeller University and a postdoc in Janelia. And now she has her own lab at the Champalimaud Foundation, where she's studying perception of self-motion and motion in general. And yeah, really excited to hear her talk about what she's currently working on. Take it away.
Thanks, Michael. It's great to be here. Thank you so much for the kind invitation and for everybody who is out there to be here. I'm gonna share. Okay. Just let me know if you can see the slide. Is that all good?
Yes.
Okay, great. Okay, so yeah, what I'm gonna tell you today about is a story that really is trying to relate the control of gaze, the control of the body movement through space, and how optic flow may be very critical for orchestrating this relationship between gaze, stability, and course control. And so before I go directly into this topic, I wanted to just bring everybody back onto the same page and really think about the importance of visual control of locomotion and what is going on as we kind of explore or walk around different places. And so let's start by this very ordinary example. Here are my kids actually walking along a new street in Buenos Aires. What they need to do as they're actually looking around and learning about different features of the city, they just need to move their eyes constantly. They need to see where they need to be placing their food. If their conversation is very interesting and if there is a particular topic, maybe they look at each other. They're doing that as their bodies are continually moving through the space. And then as you see here, there is a street interception. So as the conversation goes on and they're moving their eyes around, they might actually wonder whether they are able to cross the street or not. And so they might be anticipating whether they need to be accelerating their pace or actually hold their walking altogether based on what is the behavior of other pedestrians, other cars and vehicles, and actually the traffic lights. And we know this is all happening because of the work of Mary Hayhoe's lab in Austin, where they study how subjects, humans, are using visual information to control locomotion. And the way they do this is by asking first the question, what is the nature and the structure of that pattern of visual information that is getting into the retinas as we move through space? And so what they do is they use all possible gadgets. They put goggles that are semi-transparent so the subject can actually look around. But at the same time, the experimenters can track the eyes of the subjects. They also have accelerometers on their heads, and they cover the subject with all these motion capture items such that they can, here on the right, reconstruct all movements that are happening across the body as the subject is doing quite an ordinary task. It's asked to just simply walk around on a very easy but unfamiliar terrain. And so the reconstruction looks kind of like this. It has the 3 axes of rotations of the head. It has the line of sight or gaze, which is going to be the focus of the talk today. And it has the sort of left and right part of the bodies. And these dots that you see here are the locations where the food is going to be placed. And so from this very specific example, what I'm showing you here effectively is that the subjects very frequently actually look to particular places, retain that information there, and then use that information to plan where exactly in the short future, in the short-term future, they're going to be placing the foot. This is very important because bipedal locomotion is highly unstable, and posture control is online and working as soon as the body gets into motion. So when we look and we study kind of control of gaze, we typically think about a simplified version of what I just mentioned. What does it mean to maintain a gaze stable during locomotion is a really important question here. So first, the definitions. What is gaze? This is a reminder to everybody. Gaze in an allocentric reference frame, a world reference frame, is the sum of the eye angle with respect to the head angle with respect to the body, and the body angle with respect to the world. These three coordinations between the eyes movement, the head movements, and the body movement is what produces the direction of the gaze. Okay, so keep that in mind. And the problem about maintaining gaze stable is that there is a lot of movements that are mechanically or biomechanically constrained by the locomotive system. So if we look at the head, which is very critical in this gaze stabilization mechanism, for instance, and we look at it even at the in the context of the stride cycle. So what I'm plotting here is the head velocity of the subjects in three different axes: the anterior, posterior, the vertical, and the lateral axis, as a function of the percentage of the stride cycle. And one of the things that I want you to really look at is how much motion of the head exists that is coupled with the actual step cycling, right? Here you have differences in terms of whether the right foot or the left foot was actually the one to be placed or transitioned from swing into stance. And so that compromises quite a bit the idea of gaze stabilization during locomotion. But of course, we know that from the work that I just described, in fact, there are very brief moments in which subjects, in the context of movement of the body, maintains that gaze fixed. And we know this is based on a series of different reflexes, including the ocular motor reflex, the vestibular ocular reflex, and the vestibular collicular and spinocollicular reflexes. These are different and they're coordinating the motions between the eyes, the head, and the body. And one of the things that we are interested in the lab to understand is, first of all, the sort of complete circuitry underlying these different reflexes, some of which we know quite a lot, especially in the vertebrates, some others less so. But most importantly, we don't know how those are coordinated to actually support this idea of gaze stability in the context of walking. Now, visual information is entered by this window of time in which gaze is very briefly fixed, and then gaze is rapidly shifted to different positions. And in this manner, the subject here is able to acquire that visual information that is so important for many different aspects of walking control, from posture control by the sort of planning of where are you going to be placing those foot, from decisions that might take much more in time, such as the one that I was alluding to, whether I should be able to cross the street in time or not, I should need to stop my locomotion, I should need to turn once I see something that might be very interesting. And so if you look at this more as a function of time, here I'm plotting the sort of head and the eye components of the gaze stability in walking subjects. And so you can see how nicely these gaze, again, are stable, and in between they're very briefly shift. This is no more than within 200 milliseconds. And what's actually really interesting about this control of gaze stability is that it's highly conserved across many different animal species, including invertebrates. And I'm going to be focusing a little bit more on flies later in my talk. But what I want to actually posit as a particular challenge is that even if you have these great reflexes that are orchestrating movement across all the possible eyes, head, and body movements across the body, there is such an uncertainty in the environment that many of the planned actions might actually go really wrong. And these changes can actually happen on a moment-by-moment manner, and if you're not able to correct for them, then huge instabilities can amplify very quickly in such a dynamic system as locomotion. So to illustrate what I mean, I wanted to just bring you a very easy example. Let's assume that these reflexes are all working super nicely, and let's assume that actually in this particular moment, the subject is having the eyes, the head, and the body all aligned to the direction of locomotion. This is the pattern of image velocity that results from a situation in which the subject is actually walking forward in a straight manner. And what I'm highlighting here in magenta is really the sort of visual scene and visual motion status in this particular area, which is a fovea of our retinas, where you see what we define as gaze stability. That is, you have minimal retinal image at that point. However, if you're unable or you're rotating your body, you're not moving straight, but you're rotating, you can see very nicely that the condition of gaze stability breaks down, and now even in the fovea, you have quite a bit of visual motion information. And what's actually, this is condition, for instance, during rapid gaze shift, and potentially the reason why those are actually controlled to be minimal in time. Most often then is this particular case where the animal perhaps is intending to walk straight, but actually is not able to do so because of instabilities in the terrain, or like the status of the muscular system or muscular skeleton systems prevents for like quite a really nice asymmetric walking. And there again, you see that even in this fovea, where's the point of high accuracy, high visual accuracy, the gaze stability condition is also break. So a corollary of this is that in the context of trying to stabilize gaze during walking, what animals should do is to try to minimize rotational components of these whole patterns of visual motion that are typically called optic flow. Now, away from these fovea or these accurate zones, there is a lot of actually self-motion information that exists given by these patterns of optic flow that the animals can be using to actually try to minimize that rotational information. What's interesting here, and actually a little bit under discussed, is that if this is the case, then there is a very important relationship between course controls and gaze stability. And so this is what I want to focus today in the talk. We want to focus about this particular relationship here. Now, if the animals are using these patterns of octave flow that are actually arising by the movements of the body, and they're binocular in nature because the two eyes are actually being excited by this. And if it is indeed the case that during exploration, animals are trying to minimize as much as possible rotational components and this pattern of octave flow, here's a situation that should be a very strong selective cause for the evolution of computations within the processing of octave flow. What I'm showing you here now is a fly, which is a model organism that we're focusing on in the lab, in two conditions. In both conditions, which I am depicting here by walking path, in the left we have a situation in which the fly is walking with a certain amount of angular velocity and with a very low forward speed. And as a result of this, it generates what we know as the asymmetric pattern of octave flow, and is asymmetric because the right eye and the left eye structure of this visual flow is different. And so if you have a circuit that is integrated information between the left and the right side, they can actually pick up these differences very rapidly, and they can use that information on a moment-by-moment basis to actually correct the course of this fly. However, when the animal is walking at high speed, these asymmetries are a lot more difficult to detect. They are actually, in fact, masked by the translational component of the walking behavior of the fly, which tends to induce symmetric patterns of octave flow across binocular, across the two eyes. But still, the system should be able to detect these asymmetries, these underlying asymmetries, to minimize the potential accumulation and amplification of drift. This visual information is very important to maintain course stability and therefore gauge stability. And so this is the question that I want to focus today in today's talk. Before I jump into the data, I want to give some sort of definitions. First, what is the gaze definition in flies? So here, what we are going to be defining in this talk is gaze potentially from the mostly from the perspective of the contribution of the head rotations in relationship to the body angle. We know that flies move their eyes. They move their eyes in different ways as we do. And I think there was a talk by Lisa Feng here in this series that I'm sure she explained beautifully what is the basis of that eye movements. However, we don't still have a good system where we can actually look at the motion and coordination of the motion between the eyes and the head and the body in the context of freely walking movements. So for the moment, I'm going to be restraining myself in terms of the relationship between the head and the body. Now, as in the case of humans, in flies, and this is coming from work in the Martin Engelhardt lab with larger fly species, as well as our own Introsophila melanogaster, we also see that there is quite a bit of head velocity signals, both anterior-posterior as well as medial-lateral, that is fluctuating during the stride cycle. And this potentially is, again, perturbing gaze stability during this moment-by-moment basis in the context of forward locomotion. Previous work in the lab has nevertheless describe a particular principle for the head and body coordination in the context of forward locomotion, forward walking. And here what I'm showing you is an analysis of this coordination shown by a simple cross-covariance analysis between head movements, head in-body movements, and body inward movements. These are done using a high magnification system where we can track the flies freely walking around, and then we can use machine learning-based computer vision tools like DLC to be able to track the head, the body, the abdomen, the legs. And so what this is showing here is that there is a particular correlation. If the body is rotating, the head is following that rotation, but it's following that rotation in the opposite direction. So this is kind of very similar to what happens with the vestibular collicular or spinal collicular reflexes in us. And this is also suggesting that flies during the course of forward walking, they're trying to actually maintain gaze as stable as possible, defined from the perspective of the head-body coordination. And so what we know from the flies, again, from previous work in the lab, is that in freely exploratory flies, they actually arrange their behavior in a very similar way as what we do during exploratory walking, in that they have moments in which they're walking forward and maintaining that gaze super stable, and those are interrupted by, again, very rapid changes in the body rotation and in the head rotations that are actually happening in the same direction. So the coordination during gaze is completely different as the coordination between the head and the body during forward locomotion. And this is suggesting, again, this idea that there are these conserved systems across many different animals where during exploration and navigation, gaze is either fixed or rapidly shift. And what we also know is that flies are very sensitive to visual feedback, that is the self-generated visual motion information, to be able to control their course. And so here is a schematic of the results. You can read the data from this paper. What I'm showing you here is conditions in which we have very strong visual feedback, which means that the fly can really detect that very easily, and very weak visual feedback or darkness in which the fly cannot really detect visual feedback. And correlated with this strength of the visual feedback is the capacity of that animal to actually walk straight. So recall that if the animal is coordinating head and body in a way that is aiming to maintain gaze stable, which is in these conditions, but is actually drifting, that actually jeopardizes gaze stability, and this condition is less stable from the perspective of gaze than this one here. Meaning that in this condition, there's going to be quite a bit of rotational information, where rotational optic flow information, where here there is actually a pattern of focus of expansion that defines an area on the retina of the fly of minimal visual motion information. What we actually ask in this study is, why is it that flies are actually drifting if they want to maintain this condition here? And the answer to this question came by analysis of posture stability based on the tracking of the legs, which I don't have so much time to tell you about, but what I wanted to show you is a particular relationship that we found. What I'm actually showing you here is once posture is perturbed, I'm plotting here the recovery of that perturbation as a function of how straight the path of that fly is. And what you can see here is that this is an inverse relationship, meaning that there is some sort of trade-offs between maintaining walking with a maximal stability of the posture versus maintaining walking with the maximal possible gaze stability. And this is kind of interesting, and the color codes here, which is again related to the strength of that visual feedback, suggests that the orchestration between this trade-off is actually done by this visual feedback processing circuits. And so one cue that should be very important for these visual feedback processing circuits is visual motion information. And so if our hypothesis is correct, then, meaning that if visual motion is really orchestrating the relationship between posture stability and gaze stability, then if we generate flies that are unable to detect visual motion, the behavior should be kind of collapsed to the condition of darkness, when the flies cannot use visual information or visual motion information to maintain a straight course. And indeed, this is what we observe. So if we express an inward rectified channel, specifically in the populations of visual neurons that are processing and computing motion, we see that now those flies are all behaving as if they were in darkness, even though the visual world is changing as in the same way as here. So what is this telling us is that there is this important relationship between gaze stability and course control, and that this is actually shaping the possibility of the fly to maintain its gaze stable, and is actually regulated by rotational visual feedbacks. So we wanted to, this give us an opportunity to kind of enter into the brain of a fly and really understand what is the functional organization for these visual feedback processing circuits in order to be able to orchestrate this sort of trade-off. And so a little bit of an introduction for those who are not very familiar to the optic system of the fly. So the fly has a compound eye, which means that there is many different little lenses here underlying or covering many photoreceptors. The retinotopic organization is such that the photoreceptors that are looking at the same point in space will be converging their projections into the first layer of the optic system called the lamina, that will be forming these sort of columnar or cartridges and columnars later on that are going to be maintaining this retinotopic information about changes in luminance. At this point here and at this point here doesn't really, which is highlighted here and here, is where the motion, direction, selectivity emerges, and this is the famous T4 and T5 cells that we have been expressing the inward rectifier channel. And so postsynaptic to these cells, is a huge diversity of neurons that are very important for different actually functions. I'm going to be focusing on this population here called the lobular plate tangential cells. I'll show you in a second what those look like, but I wanted to tell you that we have these neurons located in the lobular plate, which is this fourth neuropil from the photoreceptors. And they come in different flavors. Basically, their dendrites are located specifically in one of the 4 layers of the lobular plate, and that is providing them different sensitivities in terms of wide field patterns of visual motion. They are actually tangential, meaning that their dendrites are orthogonal to the organization of the retinotopic map, and that means that they actually lose the retinotopic map and they're integrated information across these different locations in the monocular field of view. And so work with our collaborators at Genilia, the lab of mycorrhizae, doing EM electron microscopy analysis. We're able to identify about 60 types of lobular plate tangential cells, like it had been previously reported for other larger fly species. What I'm showing you here is in yellow the sort of coverage of the dendritic field of these cells. And then these little triangles in color code here is based on computational anatomy. So depending on the inputs of these T4 and T5 cells, there is like the prediction of the sort of patterns of optic flow that these various different cells will be sensitive to. We confirm by this analysis the sensitivity to a whole set of lobular plate tangential cells that have been classically characterized already with electrophysiology, and this is the focus of the talk today. So these neurons are beautiful, as you see here. They are bipolar. They have a cell soma, dendrites, which are large and elaborate, and then they send their axons to the central brain. So these are visual projection neurons. We have about 12 of them, which were already characterized. And these properties of their direction selectivity for wide field patterns can be classified across 3 axes of rotations, which are depicted here. So the blue neurons are sensitive to rotations along the pitch axis. The green cells are sensitive to the rotations along the roll axis, and these magenta cells are sensitive to the rotations along the yaw or horizontal axis. So we are going to be focusing on these magenta cells. And so these magenta cells, these rotations along the yaw axis, of course, can be induced by pure rotations of the head on the same axis, or pure rotations of the body, or a combination of the two. And in fact, in anatomical studies, we know that these neurons are really tightly related to neck connectives, really suggesting a strong function for head rotations. And for both motor neurons that are projecting directly to the neck, but also descending neurons that have dendrites on the brain and project their axons into the spinal cord analog of the insects, which is called the ventral nerve core, or VCN. And these descending neurons are projecting in areas which are controlling, again, the neck, but also the wing and the haltear neuropil, suggesting that these neurons are coordinating neck and head and body, specifically in the context of flight. And work from Lisa, actually, and Amon Kim, when they were both postdocs at Gabby Maimon's lab, has shown that if you have a flight tethered and you move the whirl, these flies are going to be trying to rotate. This is very noticeable by the abdomen, the rotations of the head, and then some sort of like left versus right asymmetries in the windstroke of these flies. And if you track these head angles and you now silence these populations of lobular plate tangential cells, you see a quite significant decrease on the visual induced rotations of the fly, suggesting that indeed these animals are actually really contributing to head rotations. Now, previous work in the lab has actually also shown in a slightly different paradigm. Here we are now having a fly that is walking on a ball, and we are performing in simultaneous recordings from one of these neurons, one of these yaw-sensitive cells. And then we are at the same time inhibiting them by the external application of histamine that is sort of opening histamine receptor that we artificially express in these cells, and that should be kind of inhibiting the neuron. And we can do this either on one side or on the other side. And then we can also do this depending on how fast the animal is actually walking. The reasons of these details are kind of can be found in this paper here. What I wanted to show you is if you have control flies where there is an expression of the ORT but not driven by the GAL4 that labels specifically these yaw-sensitive neurons, here's the activity of the neuron. You puff histamine in these dotted lines, you don't see any effect on the level of the activity of the neuron, and you don't see also any effect at the level of the behavior of the animal. And here in cyan, I'm showing you trials in which we are presenting histamine when the fly is walking at low speed, and here when the fly is walking at high speed. And these are the conditions of that stimulation. If, on the other hand, we actually are expressing this histamine sensitive channel. We puff histamine, we see the inhibition at the level of the neuron. We don't see any effect at the level of the behavior of the animal, and this is again the condition by which we presented the histamine. However, when the fly is actually walking at high speed, even though the effect of the histamine application is identical to the condition of low speed, now we actually see an effect of behavior. And these experiments have suggested that actually these yaw-sensitive neurons can actually control walking behavior, even if there is no explicit knowledge in terms of descending neurons that are connecting these neurons to leg neuropils. And most importantly, they're able to control or course control based on the overall motor context of the fly. That means whether the fly is walking at high versus low speed. Okay, so to summarize in this, we have a subset of these lobular plate tangential cells. They're very sensitive to visual motion in the yaw. They're able to control course, and by doing so, and head motions by doing so, they're able to actually contribute to gaze stabilization. So how about the physiological responses of these cells? So here is a typical experiment. We have a fly that is seeing a whirl based on these random dots moving in a particular direction. And this is, in fact, the preferred direction of these horizontal sensitive neurons, yaw-sensitive neurons. Here are two different classes, a magenta one and a violet one. These two neurons are actually sensitive to different directions of motion. So this magenta fly is sensitive to what we call in the fly progressive or front to back direction of motion, whereas this violet neuron is sensitive to the opposite direction, regressive or back to front movement. And what we know previously from the field is that these two neurons converge at an area of the brain, and that is where the dendrites of this descending neuron is located. And so consistent with the sensitivities of these both these inputs, the red neuron is highly active in the context of these rotations of the world. One thing that I want to mention, if you have to imagine that you split this stimuli in half, and so you have one side of the hemisphere being front to back motion and the other half of the hemisphere being back to front stimulation. And that's the optimal condition for these two input cells, and therefore it's not surprising that the descending neuron, postsynaptic neuron, will be activated as well. Now, this is a condition that is trying to mimic when the fly is actually walking super straight. In this case, we see activation of the magenta cell, because now in the two hemispheres, we have this progressive motion of front to back movement. So this is stimulating the same neuron, no problem. But if, of course, it's actually not stimulated anymore, this secondary neuron, because this is actually not anymore regressive or back to front, is actually the opposite null direction for these violet cells. Now, what's actually surprising is that the red neuron is also not responding to the stimulus, even though the magenta cell does. And then the condition which we want to really study here is the one in which you have these two combinations, so rotation and translation together. So here, we still have a case in which this magenta cells response, this is still front to back motion in the ipsilateral side. There is a bit of front to back motion, and this neuron still is not responding much. But now the surprise factor here is that the red descending neuron is really responding, seemingly trying to detecting this minor component of rotation. And if you look at the activities of these input cells, you would actually say that there's not so much difference between these two. Maybe there's a slightly more activity here. And that really seems to suggest that there is some sort of gating mechanisms that are happening between the inputs of these cells and the output of the system. But we have really no knowledge in terms of the actual circuit that exists once these neurons are projecting into the central brain. We knew of the existence of this red descending neuron, but we wonder, given these physiological properties, whether this was the only such neuron, and so this sort of gating mechanisms had to do with synaptic physiology properties, whether there was a lot more going on, including more neurons that could actually explain us the changes in the sensitivities of these cells relative to their inputs. And so for that, Mert Ergikaya, a super talented PhD student that is now doing a postdoc with Jan Ak, took the laborious strategy of actually using electron microscopy data sets, but this was early on, so he did most of the work by manual tracing instead of the sort of more recent modern, automatic segmentations and tracing and tracing. So he joined efforts with, again, the group of Mike Reiser, and the idea here, who have been already identified these magenta cells in the EM datasets, and the idea was to identify the output side on the axons and then ask the question, what is these neurons connected to in this area of the brain? And so then you can trace those neurons and use, again, intelligent tools to go from these skeletons to the genetic lines that we can construct in the lab, and so we can actually have direct access to them. And so what Mer has found is that there's a little bit more complication. Here is where that red descending neuron is, but as you see, there is a lot more going on. There is quite few more descending neurons, but also other neurons that I'm going to be trying to explain to you by this reorganization of the network. Still is very messy. There is a right-left symmetry. Again, the violet cell is coming from the opposite side and is converting inputs in the descending neuron that we already know that has these Kerke properties. This red neuron receives convergent projections from the magenta cell, but as well as a bunch of GABAergic neurons. And in fact, what we have described in this study is that this is a compact multi-layer network where we have a visual input. We have actually a divergent output. Some of these outputs are going to the ventral nerve cord, but some of other outputs are actually going deeper into higher premotor areas of the fly brain. And then there is this middle layer, which is actually based on recurrent interactions between GABAergic interneurons. So we do not know anything about the properties of these cells, nor the properties of these output neurons. So the next slides are going to be about the physiological characterization of these cells. So we started by performing two-photon calcium imaging of these neurons. We started by asking the simple question of, are these neurons sensitive to rotations as what the connectivity will predict? And indeed, that's the case. So we have these three different interneurons that are responding readily to rotational optic flow. If we now contrast their responses with patterns of optic flow that are mimicking translational information, as we expected from inputs from the violet neuron, but not so much from the inputs from the magenta neuron, please note that all these interneurons are actually receiving convergent information from these two visual inputs. Some of these neurons are absolutely shut down by translational information, whereas this green neuron is actually enhanced in its response. And that was quite surprising for us. And we wonder whether this was a property of this sort of recurrent GABAergic information. So in the flies, you can use these very sophisticated, beautiful genetic tools and then target the GABA input signaling, particularly and only on this green cell, and render it insensitive to information passing through GABA receptors by just really getting rid of that GABA receptor. And so if we do that, we see that now this green neuron behaves as expected by the properties of this magenta neuron, which is sensitive both to these rotations, but also to this translation information. It seems that this neuron in this condition is not sensitive to the suppression of activity that might come from reducing this input here from the violet cell. And in fact, if we do comparisons across all these different elements from this network, including these two outputs here, we notice that if we compare the activity of these cells for between rotational optic flow or translational optic flow, and we kind of come up with this discrimination index, which says, from the activity of the neuron, can you tell me, how much can you tell me that the anima have been presented with rotational versus translational information? You see that the input is a little more active, let's say, towards rotational information, but these magenta cells, there's a couple of them that actually they're completely not sensitive to either. We have this green cell that is actually really much more strongly responding to this translational, and then the output becomes even more rotational sensitive compared to the input. And so here you see a really beautiful sort of input-output transformation of the sensitivity and the selectivity of these cells to rotational versus translational optic flow. And so to start to understand a little bit how this could actually support course control, we use this connectomics information and the physiology to kind of build up a connectomics biologically constrained simple network of an agent that will have inputs these magenta violet cells, the middle layer, and outputs this descending neuron P15. And we have this agent really walk around the world, and it will be self-stimulated both with translational optic flow as well as rotational optic flow. And so we put some sort of rules here so that this agent mimics a little bit the exploratory locomotion that I've already mentioned to you, which is based on these, forward runs and then very rapid turns here. And so we started looking at the sort of activity of this left versus right descending neuron. And sort of the most, when in relationship to whether on average these cells would be reporting some asymmetries of the system or would be reporting more sort of symmetric information. And so what you can see here is that indeed the output of the cells are much more sensitive to this asymmetric report based versus these inputs. And if we silenced in the network, in the agent, these green neurons that are weirdly sensitive to translation information, this enhanced rotational selectivity is abolished, and now the activity of these output neurons are fully explained by the visual inputs. And if we repeated these experiments in physiology, where we present, again, a sort of translational information, and we have a situation where we control, in control, which we have like just a normal fly, or a situation in which the inputs of these two neurons, the GABA inputs of this neuron is blocked, then we see that in fact, indeed, is the case that if this neuron is no longer sensitive to GABAergic input, then it actually starts to respond to the translational information. And so what's actually also very important is that this model predicts that this translational sensitivity, especially for this green neuron that seems to be so critical to maintaining the output selective to rotations, even in the context of translation, is that it does so even within a regime that is biologically relevant, meaning this is sort of the distribution of forward speeds that we see with the real flies. But now the question is, can we actually reproduce the visual conditions in physiology that will be related to a situation like this? And for that, we started to present compound optic flow. So here what we're going to be doing is we're going to have a constant angular rotational optic flow, and then we're going to be adding the translational component across these rows here. So in this zero translational component, this is pure rotational optic flow. And so you can see that this is rotations to one direction, the null direction of the neurons. This is to the preferred direction of the neurons, and this is to the preferred direction of the neurons when we are actually silencing these GABAergic inputs to the output of the cells. And here is a condition that we are going to be seeing again with these two for this particular green interneuron that is very sensitive to translation. But it also responds to rotations like you're seeing here, either in control conditions or when its GABAergic inputs are also knocked out. As we are increasing a bit of translation, this is a condition in which there is a huge asymmetry on the binocular system, things are more or less the same. But things become a lot more interesting when now these translational components start to mask this rotational component of the optic flow. So in this condition, a couple of things happened. First of all, this green cell that starts to be, instead of rotational sensitive, becomes translational sensitive. So you can see in the control that there is a gate, a switch here, in which this green neuron loses its direction selectivity. However, this direction selectivity is maintained in this compound stimuli if we actually knock down this GABA input. So this is again suggesting that even in more naturalistic optic flow conditions, GABA inputs in the recurrent system of this middle layer of GABAergic neurons is very critical for this sort of gating of the interneuron to become translational sensitive from rotational sensitive. Now, if we look at what's happening in correlation with this, we see that now in the case in which the output of the system is also not sensitive anymore to GABA input, including input from these cells, now become completely identical to the responses of the visual input. And in fact, it's in the null direction, and in fact, even a bit more sensitive to the preferred directions. Now, you know that the amplitude of these responses are actually a little bit decreased. This is because all the cells are actually velocity tuned, and we're running these experiments in non-behaving conditions where the speed of the profile here, so the sum of the velocity of the rotational component and the translational component, is providing overall speeds that are already on the sort of decreased side of the velocity tuning of these neurons. And so when we increase even more the speed of the translational component, we kind of see the same trends, even though now everything is even more reduced. But what this is actually really suggesting to us is that these recurrent and sort of competitive networks, so this green neuron actually sends projections to the opposite side, is allowing the system to be able to extract the asymmetries of the binocular structure of the optic flow input that are actually masked by the forward speed of the animal. So if we run these experiments in the model again, with and without this middle layer, we see that now the system is absolutely not sensitive to these asymmetries in the input. And those asymmetries are really required and dependent on the activity of this middle layer, inhibitory recurrent middle layer. And so this also suggests that if you do experiments like if you silence one of the outputs in the agent, that would be inducing drifts on the walking of the agent. That's not surprising because we designed the system to be only depending on this red output. But if we repeat the experiments with a real fly, we actually see the same effect. And this was super interesting because it suggests that this descending neuron, even though it is projecting to neck and wing and haltear neuropils, in fact, in the context of walking, it also has an effect on course control. And if you now look at the electron microscopy data set from the ventral nerve core, we actually found that this descending neuron is connected to interneurons, strongly connected to interneurons that are actually projecting to the legs. And so effectively, we found an anatomical substrate for this important coordination between the head and the body in the context of course control and gait stability. So we're wrapping up the talk. What I show you today is in order for the systems that are processing visual information, especially related to the areas of the visual, the retinal input that are very rich on visual motion cues, there is this competitive disinhibition configuration between the left and the right eyes that are trying to extract these asymmetries that might not be super evident in the input of the system, such that they actually promote an output that is a lot more sensitive and selective for those asymmetries, even in the context of high speed of locomotion. And the actual sort of synthesis of these relationships really mimics something that has been recurrently found in the literature, this sort of inhibition of the inhibition configuration. And this sort of structure of the network has been really linked to functions that are related to simple behavioral choices. In this case, could be think of as course control to the left, course drift to the right, robust categorizations, noise reduction in the retina, and pattern completion in the context of hippocampal activity. And it actually is really remarkable that we kind of see in these different systems sort of a repetitive theme in terms of the circuit structure that are actually linking very specific simple computations that are really related to detecting things that are different between the left and the right side, and in this case, in the context of gaze stabilization and course control. The other thing that we actually found is that, in fact, the networks I focused on the talk are related to this horizontal system and the yaw-sensitive systems, but we also, of course, revealed connections, central connections for the roll and pitch. These are actually all merged together. So it's an interesting situation where we have the visual input that is very nicely separated in this axis of rotations. We go through the central networks that seem to be reducing that three axis into a two-dimensional system in somehow sort of an abstract representation of the visual motor circuits. And then again, when we go, at least in the case of the head rotations, we see beautifully, again, a divergence of multiple motor neurons that are seemingly involved in, again, three different axes of rotations of the head. And this kind of like dimensions that are, being reduced in the in a sort of more abstract representation, meaning that it's not really sort of encoding the physical axis of the motion, not until the actual actuators or effectors are at the effectors level. That's what I meant. Has been again repeatedly found in gaze control systems in the context of head-body coordination, and importantly in the context of multisensory integration. That means that there is this sort of coordinate transformation here, or dimension reduction, that allows multiple signals that are coming in different reference frames, retina, head, body, to actually be mapped together for the proper calculations and computations that need to exist. And in fact, in the lab, we have found all of these neurons are actually sensitive not only to optic flow, but they're also sensitive to non-visual information in the context of locomotion. I don't have the time to go over here, but just wanted to emphasize that this middle network specifically is a central hub. It receives really widespread inputs from many different regions of the brain, including the ventral nerve core, suggesting a really interesting bidirectional communication between the head into the body and the body back into the head. And again, so what I wanted to go back into the very beginning, we kind of go back into the sort of behavior, the exploratory behavior of a fly. It has some sort of a structure in terms of how it controls gaze that is very similar to how we do it. And this is divided into sort of gaze fixations and gaze saccades. We don't still understand how these are controlled, the switches of these are controlled, but what we are starting to reveal is that there is this important component of gaze fixation which has to do with kind of trade-off between the posture, the mechanical posture stability of the body and the gaze stability. And we have shown in this talk that the sub-network that is contributing to this orchestration has a bit of a very simple sort of classification system based on really important contribution by GABAergic information. And just if you were wondering, we also are looking at this other output of the network. And like we found with the red cell, this output is also extracting really beautifully the rotational component of compound optic flow. And we're right now trying to understand how this enriched extraction of asymmetries in the optic system is contributing to behaviors that are going to be dependent on these more higher order premotor areas. And so with these, I'm sorry about my delay. I want to thank especially the lab. The talk that the talk I talked about today is mostly based on the work of Mert, but with help of Claire Rush here, a postdoc in the lab, as well as Corina Genghart, another postdoc in the lab. And I'm very happy to respond questions if you have any. Thank you so much.
Thank you. That was really cool. Yeah, so currently there's no questions yet. Let's wait for those. In the meantime, I would have some questions.
Mm-hmm. Should I stop sharing? What do you think?
Yes, I think let's stop sharing and then we can talk about questions. Yeah, so maybe I have missed this, but everything you've shown most of what you've shown so far was in visual only stimulation, right? So I'm guessing the animals are tethered and you show visual stimuli. Do you know if do you have any idea where there would be interaction if the animal walks and how this would modify, like this proprioception, how that would modify the sensory input, if this would happen in this mid layer as well?
Yeah, so that's a great question. So that's a couple of series other questions that I decided not to talk today about. But so yes, I very briefly mentioned at the very end of the talk, both the visual neurons and these more sort of multimodal neurons in the central brain, the GABAergic neurons, all of them are actually responding to visual and non-visual information. And they do so in very specific ways. So with the flies, the visual neurons, now I'm talking about visual neurons. So if the visual neurons are, so if the fly is walking with high forward speed, presumably trying to move forward, the visual and non-visual information is actually summing up. And it's summing up in very specific ways. So there are congruent interactions. And we think about that as the potential increase in the sort of robustness of the encoding of the self-motion information. Now, work done in Gavi's lab, but also in our lab, if we now look at the same neurons in the context in which the flies seem to be doing a rapid turn or a saccade-like sort of turn, both in flight, again, from Gavi's work, but also in the context of walking on the ball, in our case, we see that the sign of these interactions changed at the very beginning of the phase of the saccade. So there you have actually suppression of the activity of these visual inputs very briefly, and then a rebound of activity that is now multimodal. And the time course of this is really nicely correlated with what we observe in terms of the head and body coordination in the context of those saccades on the ball as well. So what we see in the context of saccade-like events on the ball, we see that the head and the body are turning in the same direction initially, and then as the body is continuing rotating, the head actually starts to counter-rotate in a second phase, such that, again, gaze is quickly stabilized. Mm-hmm. And so this dynamic is really nicely matched by the sign of the interactions between the visual and the non-visual information in the cells, in the visual cells. So at the beginning, suppression, and then at some point, about 60 milliseconds after, which is the moment in which the counter rotations of the head happens, the non-visual information starts to actually be combined congruently with the visual information. So I think this is very important and actually suggests how really dynamically this system is tuned with multimodal information. Now, where are these signals coming from? So we have found, using the connectomics, we have found many pathways that are possible, some of which are actually modulating this information. But we also need to remember that many of these neurons are electrically coupled to each other, and that's actually not visualized in the connectomics data that we have. And then when you try to perturb these electrical couplings, you're modulating so much the system that it's always very hard to make a really strong mark on it. But I would say that the way in which the activity from neck motor neurons work could potentially be a contributor of the non-visual information. Is that answering your question?
Yes, thank you. That was actually really, really interesting. Let me quickly check. No second question at the moment. Then I'm gonna ask another one that is maybe not super directly related to the talk. That's just something I'm more interested in. Is like, what do we know anything about the distribution of these differently of the cells with different spatial orientation to movement across the eye, across the retina? If there's like an area that would be mostly focused towards horizontal motion, with like something like this is the case in zebrafish, it's just something like this in fly.
Yeah. So I, again, very quickly show the kind of complete set of these low lab plate tangential cells that the lab of Mike Reiser have been, analyzing in EM connectomics analysis. There, many of them, we don't have any sort of physiology yet to be able to answer to your question. What we see through Arthur's, the postdoc that is involved in this study, who developed this really cool computational anatomy tool to predict what could be the optic flow sensitivities of these cells. Many of them are actually having very structured sensitivity. So it's not like all of these neurons have a very homogeneous sensitivity to, yaw, pitch, and roll. There's various combinations. And also their dendritic organization is slightly different. So not all of them are actually super wide and spread and covering large areas. So there's some that are a little bit more narrow. Some others are really wide, like I showed you today. And again, there is a lot of information that we're still missing in terms of potential inhibitory interactions between these two that will be shaping and sculpting exactly what would be the properties from the perspective of the retinotopic organization. So all of these cells are actually integrating across large areas of the retinotopic map. I don't think there is, at least for the cells that we actually describe in the talk today, anything that points to a very specific enhancement of one particular area versus another one. In terms of horizontal aspect, there is something that is very interesting, that some of these neurons actually are coupled in the contralateral hemisphere. So there's contralateral interactions among the visual neurons themselves. And this actually provides the possibility for some of these neurons to actually have a little bit of a binocular sensitivity, so not only to the ipsilateral, but some information about the contralateral part as well. So that suggests that this very frontal area of the fly eye, which is an area that has been described to be sensitive, binocular sensitive, and binocular sensitive, I mean not based on neurons in the brain that are responding only to binocular inputs, but simply because the geometry of the fly eye with these little lenses, some of the frontal lenses actually are projecting light from the opposite, the contralateral side. So there is that overlap that exists, which could be very useful for an animal, because it actually gets double amount of light intensity, because they can detect quickly motion on the contralateral side that is kind of coherent with the ipsilateral side. So there's there is a little bit of let's say, a hypothesis that maybe that very frontal eye is a bit special. Now, whether we do see this special aspect at the level of these visual neurons that I told you today is not the most strong characteristic of the system.
Okay, good. Then there is no questions at the moment. I have more, but maybe we can shift that into the Zoom part of the talk. Thanks everyone for attending. And yeah, I'm gonna close the stream now. Bye.
And then should I stay here?
1 second. I just want to make sure. Okay, yeah. So